14th August 2019, and I'm in Congo, Brazzaville, at the headquarters of the WHO Afro-regional offices. I am here to receive WHO's Medal of Honor and recognition that is awarded to young African innovators for health. There are moments in life that are essentially exhilarating in terms of emotions. And this is one of the moments that I've experienced. You imagine me standing before the director of WHO at the global level, Dr. Tedros, and right in the midst is Dr. Moiti, who is the director for the Africa regional offices. And I'm here to receive the prize because of our relentless efforts over a span of close to 20 years to develop a rapid diagnostic test for Ebola and Marburg. Let me tell you, why is this relevant, the story that I'm telling now? Ebola and Marburg are literally generic members or sisters of the same family of viruses that we call the Filoviridae family of viruses. These viruses cause rare but highly fatal hemorrhagic fevers within remote equatorial African villages. Occasionally, we see these viral hemorrhagic fevers spread to the urban centers. And as we saw in the West African outbreak of 2014 through 2016, the pathogens have potential to cause public health emergencies of international concern. While these diseases are easy to prevent, when you are able to diagnose the disease right and isolate the infected people, the existing technology platforms for diagnosing viral hemorrhagic fevers that are caused by Ebola and Marburg are not suited for deployment at the point of care within the remote equatorial African villages, occasionally where there are no laboratories, there are no experts there. Now, here's the importance of what we are doing, or what we did, what we have done. The test we are talking about is a fast test that yields results within 2 to 3 minutes. It is easy to use, doesn't require a lot of expertise, literally designed on the platform of what we call the lateral flow tests or the paper strip tests or the litmus tests, which literally resembles the pregnancy test. But most importantly is that we're talking about a test that is affordable within the setting of sub-Saharan Africa or equatorial Africa. A test that costs within under $3. Ladies and gentlemen, how did we come to this? In the year 2000, and this is the year I was actually in my second year of medical school, Uganda experienced what was then the biggest Ebola outbreak. Over 570 people got infected, and of these, because of the high fatality rates that are associated with Ebola, we lost 270 lives. Among the lives included the lives of Dr. Matthew Lukwia, alongside several nurses. The pandemic caused social disruption in the districts of Masindi and Gulu, where the cases were very heavy, including Kampala. And yet, at the time, the best we could do was to pick the samples, move them to Uganda Virus Institute, and ship them to CDC Atlanta. You can picture it would take us a week or 2 weeks to get results. For Ebola, even a day is too long to wait, especially when the patients have progressed to the prodrome stage. These experiences, alongside many others, treating patients with HIV, and of course realizing the vulnerability that Africa has in terms of, having to wait on the West for solutions, made me rethink the reason I went to medical school. When I completed my residency in 2005, I worked briefly as a coordinator for a research program run by Makerere University. In 2006, I was lucky to receive a fellowship to do a program in bioentrepreneurship at the University of Toronto Medical and Discovery Research District. That's MassDD. I easily took up this, knowing very well that this was the path to empower me to begin working towards solutions that will help Africans. During this program, my research focus was literally studying new modalities for gene therapy against HIV. We were identifying enzymes in bacteria that literally are able to cut up the HIV genome, destroy it, with a focus of developing them as gene therapeutics. In the process, I acquired skills that could turn out to be of relevance across pathogens. In the process, when I was trying to find strategies of identifying these enzymes, of course, I sought out partnerships across North America, and we finally came across platforms that had already been developed by industry that were producing these enzymes in terms of identifying the targets. And through the process, we screened over 2,000 enzymes, and were able, of course, to identify several of them that could cut up the HIV virus. And that work is published in the African Journal of Biotechnology. But that's just the start of the story. Once I acquired this technology, began accessing global databases for pathogens, including the NCBI, the National Center for Biotechnology, which is hosted in the US. I realized we have a lot of information out there already, sequenced pathogens, Plasmodium, the mosquitoes themselves that spread malaria, Mycobacterium, the cause for TB, Ebola and Marburg, the viruses. I rapidly moved into the process of literally studying each of these pathogens. That's when we started to study the genomes, or what we call the entire genetic makeup of Ebola and Marburg. At the same time, we studied the protein makeup of these viruses. And of particular interest, we picked interest in a protein which is on the surface, what we call the spike proteins. These are more like the hairs that are on the body of the virus. We began studying these proteins, comparing the protein from Ebola species to the proteins from Marburg. We found quite interesting things. The first thing we found is, obviously, because the viruses are related, they are sisters, these proteins were essentially quite very similar. But we also noticed that there were differences between the proteins of Ebola and Marburg. And the second thing, of course, we noticed, the third thing we noticed was that even within the Ebola species, you could be able to make a few differentiation. This led us to, of course, hypothesize, come to the aha insight that we could potentially use these as targets for developing easy tests that can be deployed within the setting of Equatorial Africa. Between 2007 and 2012, we made a lot of attempts trying to apply for grants to get money, because the proof of concept experiments demand money. Validation experiments demand money. We applied to the US National Institute of Health and a program for biodefense. And what we were proposing, of course, was to build a sophisticated laboratory, but also advance these molecules. We never got funded. At that time, of course, we also reached out to the country, the leadership of the country, warning them that these pathogens could be used as pathogens of bioterror. And it was important that we develop capacity to deal with these organisms. But after some time, of course, following these failures, luck favored us. Or should it say we were prepared and, luck came our way. During this time, Grand Challenges Canada announced their seeding grants, and we applied to the seeding grants. We particularly applied to the STARS and Global Health Program, and we got lucky. We were awarded a seed grant of 100,000 Canadian dollars. And the project, of course, that we had set out to do was development of a rapid diagnostic test for Ebola. But the key thing here is that we wanted to conduct proof of concept experiments, really try to find out these targets that we have identified. Can they indeed react with the Ebola samples and give us results that we can then move to develop a test? It was impressive because at the time, of course, we were almost, I wouldn't say almost giving up, but we were at a point where, the continued failures bring this negativity around the entire process. And you begin thinking, okay, I wish I had my own money, or I wish my government had enough money to invest in this kind of things. And of course, for us, this was an exciting moment, because any young scientist will tell you that when you are pursuing science and for the first time you grab this attention of a funder, enough for them to think your idea is worth to be funded. That's an exciting moment, extremely exciting moment. We received the money, and we started off in 2013, early 2013, to do proof of concept studies. We were lucky because at the time, we had already started building partnerships with companies across the world that synthesize these molecules, what we call the peptides, but also the antibodies. Now, we are capable of doing this within the context of Uganda. But it's important to realize that for product development, if you're developing a product that you know is going to go through the global rigor for accreditation, you must use facilities that are GMP certified. And these facilities we don't have. And that's why we chose to partner with these companies. So we worked with a company called GeneCast, and we were able to, of course, produce the peptides and the polyclonal antibodies. We reached out to Uganda Virus Research Institute. Lucky for us, among the samples, that 2,000 Ebola outbreak samples that were being shipped to CDC Atlanta, some had been gamma irradiated. Now, why do we say gamma irradiation? Because under normal circumstances, the pathogens Ebola and Marburg are so dangerous that you can't work with them in an ordinary laboratory of lower safety. You must be within what we call a P4 laboratory, and there are not so many of these on the continent of Africa. Actually, there is only one functional P4 lab that is hosted at the National Center, National Institute for Communicable Diseases in South Africa. So at the time, we needed a proof of concept before we would move on to work with the live viruses. So what happened is that having access to these gamma irradiated samples would enable us to work within a lower safety laboratory because they are infected. The virus in there is inactivated through using the process of using radiations. So we were granted access to these samples, and we worked with these samples over a span of close to 1 year until about June 2014. And in June 2014, we received an email suddenly from Grand Challenges. We kept asking ourselves, why? Because our report was due in December, but they wrote to us and said, do you have any results? If you have results, can you share your preliminary results with us? We told them we're actually done, but our report is due December, and they were like, we can't wait December. We need a report in the next 3 days. So you can picture the scaffold. I had to lay everything down, asked my graduate students to lay everything down. We dived into this data. Lucky for us, the data was extremely positive. The peptides were actually very well reacting with the antibodies within the samples, and the polyclonals were also capturing the virus right. So we hurried this up and got the report together. Once the report was together, submitted it, and that's when we realized that actually Grand Challenges was interested in rapidly transitioning us to the second phase of funding, from seed funding to what we call the transition to scale funding. Now, we had challenges at this point. Transition to scale funding demands that you need to have a matching funder. The funder gives you $1 million on the other side. You need to have another funder who provides another $1 million. We didn't have this money. But, you know, because of what was happening in West Africa, the peaking of the virus, the fact that the virus had begun to jump from continent to continent, it became obvious that everybody realized that if we had a simple test that could be deployed within the communities and to differentiate between the people who are infected and not infected, it would work out very easily. I will tell you, in West Africa, because I was in Sierra Leone, we believe that there are people who had diseases like malaria and ended up in the Ebola quarantine centers because we did not have a rapid test, or the platform for PCR would take several hours, and that you wouldn't allow these people to come in contact with the other community. We got Finally, of course, through the process, Grand Challenges sent down, employed an independent monitor for diagnostics, Halteas, LLC from California. They came over, they looked at our data, we made the presentations to them, they visited our labs. At that time, we had started to partner with a small company, Ugandan company that you probably don't hear about, Astel Diagnostics Uganda Limited. We visited the facility, and of course, we passed that phase, and we were, because of the fact that we didn't have this extra matching money, Grand Challenges put down $650,000 US to start off. We used this money to produce a more refined version of the targets, what we call monoclonal antibodies. And we tested over 12 monoclonal antibodies producing over 64 sandwiches. We talk about sandwich because in order for you to produce this lateral flow test, you need an antibody that captures one side of the virus and then a second antibody that sandwiches the virus. So we produced about 12 really good working sandwiches. And we got, at this time, all along, everybody was excited. The funder was excited. The external monitors from Halteas were all excited. We were excited because we knew, oh, everything is it. We're on target. We are going to deploy the testing in West Africa. The next phase for validating this came, of course, taking it down to the P4 and testing it on live virus. We, under partnership that we have with Professor Paweska at the NICD, we were invited, we went in to work in the P4, we cultured Ebola Zaire viruses within cells, and of course, began the testing. The initial testing, nothing was working. Repeated testing, nothing's working. We went back and said, maybe there's a problem. We carried wrong antibodies. We started testing the same antibodies with the recombinant protein we had expressed in HEC cells, and they would work. But when it came to the natural virus, they failed. I'll tell you, this is one of the most frustrating moments for a scientist, when experiments don't go. Things are ringing. And then, you know, for product development, you've set milestones. The funder is looking at them. The world has expectations, because at the time we knew our product was the solution that was needed in West Africa. And it was very frustrating. Everybody looking at us and expecting what is going on. Luckily enough, of course, we had been with the monitors for quite a long time. And the funds that we had were also dwindling. They were over. It was another year of silence, everybody wondering what is happening. But I'll tell you about that 1 year. Every time I would go to sleep, my mind would keep asking, what went wrong? What is the problem? It took us a whole year to actually find out that the protein that we are targeting on the virus, the spikes, they get masked or kept, in which case they hide. In a sense, I think this is a modification the virus takes up to protect itself from direct attack. And that's when we began realizing, for this to work, we need to find a way of removing the mask or the cap. So, 2017, we applied for a European developing country clinical trial partnership with aim of trying to develop these platforms. The same year, next year, 2018, we won an emergency Ebola grant, and that was aimed at literally validating what we call the buffer system that we need to wash off. If you have used a rapid diagnostic test based on the litmus test of lateral flow test, you put the sample on the test, on what we call the sample pad, but you have a small buffer that you add on to enable the sample flow on the test. And that's what we are trying to work with. It was, I think, this start November, December, we began getting hits, 2018, of buffer systems that could actually wash out and enable the monoclonal antibodies. And at this time, of course, my graduate students who had been working with me were all excited because it was like reawakening the dream that we had been pursuing for quite a long time. Everybody was, and one of the students says, hey, but WHO, they have announced an innovation challenge, and we believe this is what you need. We're going to put in an application. It's like, fine, no problem. So they put in the application. I sent the application, and out of 400 applicants, 30 of us were chosen. And we went to Cape Verde, the 30 of us, for an interministerial meeting of the WHO Afro region. And at that time, we had to display our prototypes. And I carried my prototypes. I'm not sure whether you have anything of this, but this is a sample of what I walk around with. I was supposed to bring a bigger portion of it, but I would like you to look at this. These are the tests, and we have a sample pad, testing test line, and then a control line. So I went with this. Little did I know that some of the people who were there were actually people that were in the outbreak, responding to the outbreak in West Africa. And this is how we emerge as the best. This is how we get chosen as the best under the category of product development. Now let me tell you something. It is very difficult sometimes to explain emotions to other people, especially when you've been pursuing a goal for a long time. And this is why I'm telling, at the start I said, okay, the experience is exaggerating, but at the same time, of course, the relief that comes with it. Because for us, when we set out to do this, the whole world knew we are doing this, and our failures would literally bring the same frustration to the entire world. And that's why at the end of the day, the excitement for us came more as a relief, because finally we knew we have a product that will be deployed on large scale in sub-Saharan Africa. Now, the other thing I will tell you is these pathogens are classified Class A pathogens. Before we did this work, Uganda was having rampant outbreaks, and we kept saying, is somebody walking in with these pathogens trying to frustrate us? And when we talk about biodefense or biological defense, it's unimaginable. But this is the next phase of, a lot of things. And it is important that we invest. Now, everybody asks, why do we do this? I think that I set off on my path largely with the aim of inspiring other scientists, younger people. Africa lagged behind in the Industrial Revolution. At the time, we were being sold as slaves, and we can't blame ourselves a lot. But even after then, Africa lagged behind still during the digital revolution. Today, we are undergoing an artificial revolution. And alongside it is a biotechnological revolution because of the availability of a lot of data, what we call genomes. My specialty is human genetics. I trained at the NIH, and I am a consultant genetist. I sit in Mulago and see children who are born with genetic abnormalities. But I'll tell you, my research focus, on the other hand, my lab, and the groups and the students are trained, our focus looks at the DNA makeup of pathogens. Why? Because we know infectious diseases remain a big problem for Africa. And unless we resolve this, I'll tell you, our budgets, our health systems will not be able to deal with the emerging pandemic of non-communicable diseases. And this is why we've really spent a lot of time in this direction. If you look at my work, I will tell you this is not the first thing we are dealing with. We are developing TB diagnostics. More recently, we've gotten funding to develop COVID-19 diagnostics. We're developing an HIV therapeutic, what I've talked to you, gene therapy. But why do we do this? Because we believe it is time to build a critical mass. Does Africa have to reinvent the wheel? You keep asking. We can buy drugs from elsewhere. But the truth of the matter is, I believe that there is technology stuck in the wheel that we need to acquire if we are going to move to the next phase. Ladies and gentlemen, thank you for listening to me. I wish you a good evening.